Good morning, everyone, and thank you so much for joining us here today in beautiful Vancouver. This is my third forum. I've been with Kidney Cancer Canada now for just 3 years, and it's gotten bigger every year that I've come, so it's fantastic that you're taking the opportunity to learn from each other, learn from some of the top researchers in Canada and physicians in Canada, so I hope you make the most of it. My panel, I'm really excited about because I have a passion for research. I think a lot of people do, but they probably don't know what it means for them. And this is one of the reasons why we felt it was really important that we bring forward some of the up-and-coming stars in Canada in kidney cancer research. This is where some of the money that we fundraise goes to. And it's important to see how research can benefit all of us, whether we have kidney cancer or not. I came to Kidney Cancer Canada through policy making. I was in charge of a national organization that looked at new cancer drug therapies and would give advice to governments on what drugs to fund. And, evidence, Canadian evidence, whether it be observational, whether it be through our randomized controlled trials, it's so critical to decision makers and how they decide whether to fund something or not. And so being involved with research, funding research, or participating in a clinical trial is so important. I can't stress enough for you how much you should think about that along your journey. I know you have many things that you're thinking about, but it's something that you should explore with your care team, because there's many ways to participate in research, not just funding it through our organization. So I'd like to call upon three leading or shining stars in our Canadian research landscape. Dr. Sean Didani, Jack Bacon, and Lucia Nappi. Please join me on the stage and we'll learn about your research. Okay, so you get to go first, Jack. So tell us a little bit about yourself in terms of your journey as a researcher, and then we'd love to hear about your work.
Sure, great. So yeah, like Mona said, my name is Jack Bacon, not a doctor, but I am a PhD student here in Vancouver at the Vancouver Prostate Centre. I work with Dr. Alexander Wyatt. Since I'm going first, I feel like it's appropriate to give just a little bit of background because I think science sometimes feels a little bit inaccessible to the general public. So what I really want to hit home and remind you all is that at its most fundamental level, cancer is a genetic disease. And what that means is your tumors are driven by alterations and damage to the DNA that's causing cells to grow and function kind of irregularly. So with that in mind, the research that I'm doing here in Vancouver is based on a discovery that was made several years ago now that showed that when cells die, the DNA inside them isn't actually destroyed like we used to think it was. What actually happens is, and if I can give you an allegory, basically when cells die, their DNA is run through what you might think of as a cellular paper shredder. The DNA gets broken down into small fragments, like small incomprehensible pieces, but the DNA isn't actually destroyed. And so when that cell degrades and it finally dies, all those pieces of DNA are released into the bloodstream. So I've got this cute little pastel image up on the screen for you, which I think really helps explain it. We've got this population of blue cells at the top. Those are your healthy, normal cells, just undergoing normal cellular functions. And when they die, they release healthy DNA into your blood. But at the bottom, we have a population of malignant cancerous cells in green, and they're undergoing a very aggressive cellular life cycle. They're dying very quickly, and they are releasing circulating tumor DNA into your bloodstream. And what we're able to do in my lab in Vancouver is we can just use a simple blood test to extract all the DNA circulating in your blood. And what we can do then is use next generation sequencing technology to separate the healthy DNA fragments in your blood from the tumor DNA fragments in your blood. And we can feed those tumor DNA fragments into powerful computers and actually regenerate the tumor DNA. So we can recreate your tumor genome just using those fragments of DNA that were in your blood. And this is really impressive technology because what it allows us to do is use that information for personalized medicine. So I know most of you will have heard that word because it really is sort of the hot topic or the buzzword in oncology today. And what it means is we're just using personalized information about your tumor, like your DNA signature, to inform how we treat your cancer and what drugs we give you. Because if we know anything, it's that we know that no two cancers are the same. And so we can use this information to guide that. And what's really important is we can use it in lieu of a traditional tissue biopsy. So anyone in this room who has kidney cancer or who has had it will know that a kidney biopsy is hardly a pleasant experience. And so we in the field call this a liquid biopsy. And it's an alternative to traditional solid biopsy in that we're able to get all the same information just using your blood. So that's what I do.
Great. That's very exciting. Very, very exciting. We're going to move on to our next speaker or our next presenter, Dr. Dadani. If you could introduce yourself as well and provide us with an overview of your research. I think this way.
Okay, so thank you for having me. My name's Sean Dudani. I'm a medical oncology fellow from Calgary. I work with Dr. Heng. Some of you may know him through this organization. He asked me to send his regrets for not being able to make it today. So I'm going to talk a little bit about the research that I've been working on, which has been partly funded by Kidney Cancer Canada. The theme of our project is similar to Jack's project, which is it involves precision medicine or personalized oncology. They're all kind of synonyms. And I think from clinical experience, and I think you could hear yourself from the panel members that were up here just prior to us, kidney cancer is not a uniform or a single disease, even though it's all called kidney cancer. In other words, there's quite a diverse spectrum of behavior from the cancers that we see. Some patients, for example, may have quite indolent, slow-growing disease, which does not require treatment and urgently and can be monitored with active surveillance, while other patients may have disease which grows quite rapidly and needs urgent intervention. Similarly, some patients might respond to one treatment and others to another, and we don't yet know exactly what explains all of this. So instead of thinking about kidney cancer like this, where kind of all patients have a similar name, kidney cancer, and similar disease, we actually, in our minds, think about it like this, where there are multiple different groups of patients who all have different cancers that behave differently, and our goal is to be able to categorize them and group them based on specific characteristics of their kidney cancer, which allow us to direct treatments specifically. So for example, just as an example in this slide here, maybe patients in the green group require pill treatment with pazopanib that we heard about earlier, or sunitinib, or other cousin drugs. Perhaps those in the purple group should be treated with immune-based treatments such as nivolumab, which we also heard about earlier, or other immune treatments which are hot in kidney cancer. Perhaps people in the yellow group have slow-growing disease and may be able to be monitored. Some patients, maybe in light blue, require surgery or would benefit from surgery, and maybe those in dark blue would require a combination of pill-based treatments and IV treatments. So the way we're planning to work on this research is to use kidney cancer specimens, starting with patients from Alberta. We have about 400 kidney cancer specimens, and we're hoping to look at those specimens and see how the patients did, and if we were able to correlate any findings, protein expression in those kidney cancer specimens that may allow us to group patients like this and give treatment in a more personalized approach rather than a kind of one-size-fits-all, which is generally what we're doing at the moment.
Great, thank you. Also very exciting. And we'll have opportunity to ask these researchers some questions in a moment. But before we do that, Dr. Lucia Nappi, if you could again introduce yourself and provide us a summary of your research.
Hi, so I'm a medical oncologist, and I'm also a PhD, so I'm a clinician scientist, which is a new kind of position for doctors who have background also in basic research. So essentially what I do is to go back from patients to bench and from bench to patients continuously in a bi-directional way, just to find out new things from patients that can be validated on the bench and vice versa. So this is my job. I work with Christian Kolmosberger. I've been working with him for almost 4 years now. Tried to transition to a staff position at BC Cancer Agency. So we got a grant in 2017 from Kidney Cancer Canada, which is the project that Jack is working on, to try to understand, first of all, if we can detect circulating tumor DNA in patients with kidney cancer, because it's true that ctDNA is detectable in many tumors, but we don't know if it is detectable in kidney cancer patients. So that was the first question of that project. And the second application of circulating tumor DNA in the project that Jack is working on is if we can detect circulating tumor DNA, can we actually have some mutation that is actionable? What does that mean? Can we use that information to propose a target agent, a drug that can target specifically that alteration? And this is like what we do on this project. Another part of this project was surprisingly positive for us. We didn't expect to find these things, but we did. Is that kidney cancer is not really considered there are some syndromes that are associated with familiar kidney cancer, but it's not that common compared to other cancer like breast cancer, ovarian cancer. So we also check germline mutation. This means the mutations that are inside the DNA of all the cells of our body that can be transmitted to kids. And we found a higher fraction and percentage of mutations in patients with metastatic kidney cancer compared to what we historically knew about these. And why is that? It's possible that our technique now are better compared to before. So we are just able to find more things because we improved the detection rate with our technique. And it's also possible patients with more advanced disease are enriching this population of patients with this kind of alteration. What does it mean in terms of treatment or practicality for the management of patients? It's possible that we can go through a more personalized treatment, and if we confirm the presence of these germline mutation, we can actually use that information to develop new treatment. And the future is very exciting because it's not just single agent, but the combination of different new agents based on the specific abnormalities we can find in the DNA. It's the future, I think. So we are working on that aspect too.
Wow. Just some really amazing areas of research. So I'm going to ask each of our researchers some questions, but please feel free to think of questions yourself. I'm sure these folks have a depth of work that they do, so they're just scratching the surface, and I'm sure they'd love to talk more about their research. So I'll start with Jack. The idea that we may not have to have patients undergo biopsies is very tantalizing. How far do you think your research, and it's sort of going to lead to another question, but how far do you think we are until there will be a time when we don't necessarily need to have biopsy for patients? And the second part to that question, or really where it comes from, is a lot of times I hear, I heard this really exciting news. It was either in the news or, on a patient forum, I heard about a new research study, but it just really seems to take a long time for that result to come and impact me. So maybe you can also explain a little bit about, why the process, why do things appear to take a long time?
That's a really good question. So from the biopsy piece, which is what I do, I'll tell you it'll be a while. So the way circulating tumor DNA works is it doesn't work in all people. So we know that it's used fairly routinely in some other cancers. So it's used very commonly in lung cancer and colorectal cancer, and most importantly, prostate and bladder as well, the other big genitourinary cancers. But the problem is we know it doesn't work in all people. And so sort of the second aim of our project is, so like Dr. Nappi said, we're looking at can it be done? And then if it can be done, who can it be done in? So we know we'll never eliminate biopsies for everyone, especially because the treatment really only works in people who have high burden disease. So it could never be used in lieu of a diagnostic biopsy, because people who have low burden disease tend just to have so much normal DNA in their blood that it just completely drowns out any tumor DNA, and we wouldn't really be able to find it with modern technology, though we are getting better at doing ultra deep DNA sequencing. So maybe in the future that will be an option. But really what we use it for is monitoring. So like we were talking about personalized medicine, cancers are dynamic and they can adapt and they can evolve over time. So many of you may have faced resistance challenges to drugs before, and that's because your tumor DNA is able to confront the therapy it's being given and adapt and evolve to overcome it. So what doctors need to be able to do is monitor your tumor DNA over time and in real time. And doing that with a solid tissue biopsy is very difficult, because surely you don't want to be receiving solid biopsies every few months. So with this technology, it'll help us monitor people's DNA over time easier and less invasively. And the reason things take so long is because, like Dr. Dudani said, the population is so diverse. So just because it worked in 40 people doesn't mean it'll work in 50 people or 500 people or the entire population of Canada. So we usually start small. My current study is on 52 people. And if it shows promise in them, so we're in our preliminary stages, but I do have a poster upstairs in the Knowledge Center, which Stephen will tell you about, and you can come and check it out. And we see it works in 30 to 35% of people in our preliminary analysis. And then we'll make it bigger, and we'll see if there's anything unique to that subgroup of people that seems to make it work. And that's really how these things work. We find out who it works in and why, and we sort of just narrow it down until we're ready to bring it to the general public.
Great. Thank you. That's always helpful to understand that there is a process in scientific research as you formulate hypotheses and then actually go through a process of sometimes many years to test that hypothesis. Dr. Dudani, you spoke a lot about the heterogeneity, really, of kidney cancer. And most people think that they think of kidney cancer as one diagnosis. So hopefully we're spurring people to be curious about what they can do to help improve the pool of information we have on the different types of cancers that actually occur. How would you recommend someone speak to their oncologist or their oncology team about getting involved with research? I know it's a little bit different than actually talking about your research, but here at KCC, we really do stress the importance of having that conversation. It may not be right for everybody to get involved with a clinical trial, either for diagnostic or research or therapeutic purposes, but it's something that you should always be exploring. So how would you suggest people have that conversation?
So thanks for a great question. I think it's a really important question, and I actually think it's something that everybody in this room and Kidney Cancer Canada can really help with. Research is incredibly important for advancing the field and allowing patients to experience better outcomes tomorrow than today, or a few years from now than today. But also on a personal level, it allows patients of today to receive the treatments of tomorrow today while they're being studied. But also, I think that's very important in the Canadian healthcare system, where access to treatment in a publicly funded system often takes time to find that funding, and sometimes we aren't able to fund all of our treatments, so all the treatments we'd like to have, I should say. So I think that it's really important for patients to engage in research, and and working with patients, we do sometimes hear, when we approach them about a potential clinical trial option, some of you may have felt like this, like we hear responses like, I don't want to be a guinea pig, or I don't want you to just test things on me, or I don't want to have a placebo drug, etc. And those are all very valid points. The first thing to say is that ethically, we wouldn't be able to do these studies and give somebody a drug that we know is not effective, or give someone a placebo. But aside from that, I think that everyone here in the room can really spread the word about how important research is. I think if a patient hears from another patient the positive experience they had in a clinical trial, or the benefits that they perceive, is very different than hearing it from a healthcare professional who some people may believe have alternative goals, etc. So I think really spreading the message is very important, and the only way we're going to get any better tomorrow is by doing research today. And there are many new drugs which have been recently approved over the past 10 years. There's been explosion of new treatments, and it's all thanks to research. So to summarize, I think it's very important, and I think everybody here can really help spread that word, because there is some anxiety around it, and I think hearing it from a patient is very different than hearing it from your doctor or your nurse or a healthcare professional.
Yeah, great. And I would also just maybe add to that, thank you so much for kind of dispelling some of the, I guess myths or some of the common concerns people have. On the Kidney Cancer Canada, Cancer de Rennes website, we do have access to ongoing clinical trials in Canada. So I would also encourage folks to look at the website and use that maybe as a starting conversation piece with your care team if they haven't brought that opportunity forward to you. Dr. Nappi, so you kind of teased us a little bit with bringing forward the idea that there could be a screening possibility in the future for genetically linked kidney cancer. Could you tell us again how far off in the future that might be? And also, one thing, again, I hear quite a bit is, why in kidney cancer do we not have a biomarker like we do in some of our in some other cancers? Why is it so hard to find a biomarker in kidney cancer?
This is a very complicated question.
Sure.
So yeah, it's true. We don't have a good biomarker in kidney cancer, and it seems to be harder than other tumors to find one. For example, for immunotherapy, in other cancer like melanoma or lung cancer, we have some, at least hints that there are some genomic or protein expression that we can look at and predict to a certain extent the response to immunotherapy. Kidney cancer seems to be completely different from all the other cancer. So we know that immunotherapy works in a certain percentage of patients, but so far there's been no biomarker that has been correlated with response to treatment to the extent that we can use it to select our patients. So it's very complex. There are a lot of groups in the world who are focusing on this research field. It's possible that the immune stimulation of kidney cancer cells happens through completely different mechanism compared to other cancer where immunotherapy is used. There are some proteins that have been associated with response, so there is a lot of research going on that aspect. Why is that different? I don't know. I don't have an answer. So we are still working on that, and hopefully we can find something soon. To answer to the first question, so the germline mutation, there has been a publication I think at the end of last year from a group in New York who reported this higher percentage of germline mutations patients with metastatic kidney cancer. So in that publication, patients with non-clear metastatic kidney cancer had higher percentage of germline mutations as expected, because they are slightly different from patients with clear cell. But also patients with clear cell metastatic kidney cancer had higher germline mutations. So this is important because if this is confirmed, of course, like when you do this kind of analysis, you have always to put in context the population that you analyze. So the population in New York, it's probably different than the population in Vancouver, and it's different population in Toronto, just because like there are some very small groups that are overrepresented there compared to here. So it's hard to apply that percentage of patients to our population here. But in general, what we know now, and our study confirmed this, is that the percentage of patients is higher than that we thought it was. And the consequence is that if there are genes that we can target with therapy, we can actually use that information to offer new treatment to our patients. Is the percentage high enough to propose a screening? Probably not yet, or we don't have data that are robust yet to support that, because this means a higher cost, like if we want to screen all our patients. But this is a very good field for research, so we can actually enroll patients in these research projects and try to answer to this question. And try to contribute to understand if that is the case. But it is very interesting, and also the genes that have been found are not the classic genes that we were thinking were associated to kidney cancer. So it is very interesting, and it's a very developing field, so it's changing continuously.
Great. And just goes to show that when you think you know enough, or you know some information about a specific disease, there's always way more to learn. So we hope that we can continue supporting researchers like you in answering some of these really important questions. I'm going to open it up to the floor to see if any of you have questions. We have obviously our panelists, but we also have quite a few folks in the audience who are also researchers. So, you know, the more that we can share and get you excited about research, the happier we are. So please.
Thank you. Can you say anything about what any kidney cancer that is hereditary?
Jack?
Now I can. So that's a big question. What specifically would you like to know?
Well, I looked online.
As you should.
Yeah.
I was just curious because I hadn't really looked since I've been dealing with my kidney cancer. And it did say that some proportion of kidney cancer, and what I have as type 1 papillary, could be hereditary, but then I couldn't find out anything more about it.
Okay. Yeah. So, so like Dr. Nappi alluded to, there are 10 characterized hereditary kidney cancer syndromes that we know of, and they're associated with 12 distinct genes. And so usually this is up to your doctor. So if you do feel like hereditary cancer might run in your family, it usually is characterized by being diagnosed at quite a young age. So the average onset of kidney cancer is about 70 years old. So if you're diagnosed in your 30s and 40s, this is usually the kind of patient population that gets screened for that. But you should run it by your doctor and say, you know, I'm just curious about this. And then if your doctor does think you're at risk of a hereditary cancer syndrome, you can be referred to what's called the Hereditary Cancer Program, at least if you're in BC. And then you're sent to a special division of the hospital where they can do genetic testing on you and see if you are indeed affected by that. But it is a fairly small proportion of people. So from the general public, it's not usually a risk. But being diagnosed young and having family members who have kidney cancer, yeah. So you should, by all means, talk to your doctor.
For my daughter, I was thinking, what do we need to know, right? Well, I will talk to my doctor. She's sitting in the panel, and the other one's sitting over there, Dr. Coleman's-Berger, so be prepared.
I think we had another question back here.
Yeah, Dr. Dudani, good to see you. We're from Calgary as well, and Dr. Danny Hang, as Nancy was on the earlier panel. But being in BC, it may be for Dr. Nappi or the team up there. I'm curious about the It's called Personalized Oncogenomics. It's for BC Cancer only on the POG program. And also Stephen Yip was working with Dr. Heng, but is out here as a co-investigator. I'm just curious, because you're talking about different sequencing, and the POG program is an experimental research program, and I was just really curious on that with sequencing and decoding the genome and DNA and RNA.
So
And if it'll come nationally.
I'll answer about the POG. So the POG is a BC Cancer Agency program, and it's essentially very similar to what Jack is doing, but starting on a different sample. So Jack is doing the analysis and the sequencing of the circulating tumor DNA starting from the blood. The POG needs a biopsy, a fresh biopsy, so needs tissue to have the DNA sequenced. Okay, so this is the biggest, like the first difference. The second difference is that how many genes are sequenced in the POG compared to the ctDNA. The POG does the whole exome genome sequencing. This means that they sequence everything. So the process is long and the cost is higher. And there are like limited resources, so it's for all patients, not just kidney cancer patients. So it's still a possibility, it's still active at BC Cancer Agency, and will give some information. At the end, the information that we get from POG are always used for the same aim, and the aim is to find an actionable mutation that we can target with drugs. So that's the final goal of, regardless if we use POG, if we use circulating tumor DNA, if we use a panel of genes that we target. So it's still a research project, so it's not standard of care. We don't really know if we'll get enough information by doing this whole sequencing to have some proteins that can be targeted by drugs. So it's still like a research project, but it's still an option in patients, even patients with kidney cancer in BC.
Yeah, thank you. I just thought it leads to Dr. Dani's part about personalized targeted therapies for smaller groups and gets you to quicker the right drug or whatever process. So, thanks.
I agree.
I think we might have time for one more. Stephen, is that right? One more question? Yep. Gentlemen.
I'm not sure if this is off topic, sort of, but I just wondered where ureteral cancer fits into this discussion.
So I can answer that. So I'll start by explaining what the ureter is. It is a tube which connects the kidney to the bladder, and the short answer is it doesn't really fit in with this discussion because it's a different cancer, but sometimes it does fit in because the ureter starts right up beside the kidney, and sometimes on CT scans or ultrasounds, we can't tell if a cancer is starting in the kidney or the ureter. And if it started in the ureter, it's treated much differently, much more like bladder cancer most of the time rather than kidney cancer. So this is the reason that biopsies are important, so that we know that we're treating the right thing.
So we'll have to end the session. I'm really happy to see you have so much passion for research like we all do up here on the stage. And I encourage you to catch these researchers. They're going to be still here for I'm thinking for the rest of the day, but also we have in our Knowledge Center, we have their research posted. So please do go up and visit and make sure you continue to ask questions and continue to ask your care team how you can actually personally get involved with research. So thank you to our panel. Thank you.